I'm gonna give sort of an overview similar to the way people gave before me about why it's actually important and timely to generate a human tumor cell atlas, how we're going to go about this, and the efforts across the community to actually do that. Okay, so we all know that tumors are very complex ecosystems. They are made out of malignant and non-malignant cells. So, for example, we have immune cells, which we care deeply about, and many other cell types which we also care deeply about, for example, fibroblasts and endothelial cells. And this diversity has been very challenging for functional cancer studies, and it's also been very challenging for finding different types of therapeutics for cancer. So now with the advent of single-cell genomics methods for dissociated cells as well as cells in tissue, we think it's a great time to move towards generating tumor cell atlases. And to do that, we actually need to bring the community to all be on board. In addition to looking into the human samples, we're also going to look at mouse models. These could be very advantageous complementary models to look, for example, at function. It would be very interesting to also look at samples from clinical trials. And of course, we would use the normal human atlas results as reference for all of these tumor types, because you can get tumors pretty much all across the organs in your body. So how do we actually build a tumor cell atlas, right? How do we go about that? So there's some sort of flow, right? We start with actually recruiting patients and getting their consent. Then we need to get the material from the patients. And in order to do that, we have to have very robust SOPs. So these could be either resections, these could be core biopsies, these could be fine needle aspirates, and they can also be different types of fluids like ascites and pleural effusions. We need to come up with a great plan for our experimental design. We need to have good tools, experimental tools for both the dissociated cells and also to look at cells in tissue. And at the end of the day, we also need to have computational tools to put these all together and to feed back in an iterative way into the experimental design. So tumors actually pose specific challenges, right? So first of all, we are dealing with tissues from live donors that have disease. And this can affect the patient recruitment, the SOPs, and the experimental design, and so forth. In terms of single cells and single nuclei, we want to be able to have access to frozen and FFP material, which is most of the material for many of the tumors. And sometimes it's very important to actually have this. In terms of spatial analysis, there's a big challenge in connecting back to pathology. We need to understand where exactly the tumor came from. And on top of it, most of tumors are also idiosyncratic. That means that they're non-canonical. So if you take a tumor for one person, it will be very different than the other. And in terms of computational tools, we need to figure out what are the epigenetic states between all the cells, and also what are the genetic mutations that arise within the malignant cells. So with generous support from the CZI, we recently launched a project that's called the START project. And this project actually aims to benchmark single cell and single nuclei RNA sequencing for tumor samples across different labs and across different methods for single cell and single nucleus RNA-Seq. And here you can see all the community members of this project. There are about 12 of them. They span, I think, approximately 9 different institutes, and they come from 5 countries. And this is a very tight-knit community of researchers. And here you can see that we're already starting to work, and we have been starting each one in their own lab to actually process different tumor types, including gliomas, breast cancer, lung cancer, esophageal cancer, sarcoma, and CLL. So this is one of the main things that we would like to do, and then we will do a crossover where we send samples to from my lab to your lab. You process with the same method that and then we compare. And one of the important things is to come up with figuring out where the variation lies and come up with methods that could actually correct them. So one of the most important things when you actually look at tumors and human tissues in general is to come up with very robust SOPs for obtaining tissues. And we've worked on it extremely hard with both of our DFCI and MGH, which are two of the main hospitals in Boston, to really come up with a very good and detailed plan. And here you can see our detailed SOP. And this actually, in order to do that, we need to have tight communication across the people at the hospital, which includes the CRC, the clinical research coordinator, the clinical oncologist, the pathologist, and the surgeon. And we need to know ahead of time what the surgery schedule is, so we can actually line up all the people also in the lab in order to get the samples processed in time, because we want to do it as fast as possible. And we also need to know how are we actually allocating the tissue. So we really need to have this in place, and we're very happy to share this with other people in the community who are actually procuring tissues from hospitals and get feedback and so forth. So please find me afterwards if you would like to. We'll also, of course, leverage all the efforts that are happening across the Human Cell Atlas. Aviv mentioned in her talk before 2 different efforts to benchmark single-cell RNA sequencing method, and we will definitely learn lessons from them as well and sort of be in contact. So one of the most important things that we can think about in terms of tissue collections, especially for cancer, is to look at nuclei preps, because let's say you have a very rare cancer type and you can get only 5 first samples a year, right? It's not as useful to you as you would like it to be. So it's very good to have this method available. You can use archive tissue, you can use tissue that was frozen in one place and shipped to you. And you can also look at very minute amounts of tissue, which is also important. So in the last year and a half or so, we've been trying to figure out and actually to optimize different conditions for nuclei prep. And in the human realm, we've been working very, very closely with Kristen Ardley, who's here in the audience, and who is the champion of all GTEx samples and pipeline collections, to look at these collections and figure out what are the best conditions for really getting good results with nuclei. So here you can see one example where we look at prostate samples, and we look at 4 buffer conditions, and you can see that with some buffer conditions you get more representation of the cell types versus others that you get less. And we've been working very hard with Kristen and with people in our lab and tested it across different tissues in GTEx, and it will probably apply also to different tissues that you're using. And for each one, we find sort of the best condition or conditions. We look at the clustering, and according to that, we continue to process these tissues, and we've done that across many tissues now, including cancer tissues, and we're very excited about that. We're also developing a toolbox for spatial transcriptomics, right? Because it's really good to have the dissociated cells, but you also want to have the spatial information. And spatial transcriptomics is a method that really actually gives you quantitative analysis of the transcriptome with spatial resolution in 2D. It doesn't give you single cell resolution at the moment, But if you take it together with the single cell profiles, it's actually very informative, and it can tell you, first of all, if you're missing representation of specific cell types after you do dissociation. It can tell you what are the cells in the neighborhood within a specific area of tissue. And it could be very important to actually figure out if you want to make probes, for example, for using multiplex methods later on, such as MERFISH. So if you combine these together, you can have a very strong approach. And the nice thing about spatial transcriptomics is that it's not high throughput, it's medium throughput, and it can be used in pretty much, I think, most labs today. So you don't need very as much specialized equipment. And in addition, of course, we're working with Xiaowei and Gary to bring in additional spatial methods such as MERFISH, Codex, and maybe. So why is it important to actually have a tumor cell atlas? Well, I'll give you 2 examples from our community, similar to people who talked before. And the first example has to do with immunotherapy, which we know is a very revolutionized therapy for cancer patients. However, we also know that some of the patients actually don't really benefit from immunotherapy. They're either resistant from the beginning or they develop resistance. So in order to try to address this question, we looked at 15 untreated patients and 15 patients that were non-responders to immunotherapy, meaning they received immunotherapy and they didn't respond. And from these 2 data sets, we collected single cell data using our pipelines. We have about 10,000 cells and 8 cell types. And the first thing that we wanted to know is, are these 2 groups different, and which cell types specifically are different? So when we looked closely at that, we found that actually the T cells, which we initially thought would be very different between the 2, are actually active in the resistant patients. So, well, we didn't find anything with the T cells, so we then looked at the cancer cells. And we found that the cancer cells from the resistant patients have a very specific signature for immune evasion, and also for what we call T cell exclusion, where they don't actually let the T cells into the tumor. So great, we have the signature. Is it important or not? We wanted to figure out if this resistant signature could predict any clinical responses to immunotherapy. So we used the signature and looked at a different data set and could actually find that it really correlates with response. These are samples that were taken from 104 patients before the treatment, and then there's follow-up after treatment, and there's very good correlation, so we're very excited about that. Okay, but immune cells do matter. In a second example from immune cells from Dana Peer's lab, she looked at immune cell landscape in breast cancer. She looked at 8 patients collected from both tumor and adjacent normal. And she also looked at the peripheral blood and lymph node. And they looked at about 47,000 cells. So Dana developed, and her lab developed, a very powerful clustering and normalization tool to overcome batch effects, and it's called BISCUIT. And when they used that, they found that the tumor and normal exist are very similar to each other, actually. And the tumor looks like an expansion of the normal tissue. They also saw that the tissue resident cells within the breast are very different from the ones that were found both in the blood and in the lymph node. When they looked more closely at the T cells, they could define a continuum of states, and most of the variation was captured by a few axes of variation, including hypoxia activation and exhaustion. And what this tells us is that in order to really understand what goes on in tumors, we have to look very careful at the TILs. We have to look at their state in a very defined way. And in most cases, this might be a continuum, so it's very worthwhile not only looking at clustering methods, but also looking at diffusion maps. So I tried to give you a brief overview of how we are thinking about building a tumor cell atlas. We are very open to having as many people join this effort as possible. We're very excited to expand our collaborative networks. There are several ways to do that. One, there is currently a Slack channel for the tumor effort. And actually, I didn't tell you before, but this effort actually sprung from one of the lunch meetings that we had in the Stanford meeting for the technologies. We just all sat together and we were like, let's do it. So I think that's something that could be said about our community, that people are very proactive and get things done. We've also set up specific channel in Protocols.io for protocol sharing. The first email I got once we got the funding was like, okay, I want to upload my protocols so we can all look at them together. And I think that was super exciting to us, and we're very happy to share across the community and help. And in this case, even sharing the protocols for the normal tissues, I think will be very helpful for cancer. And last, you can always contact Mario Suva and I, and we're happy to connect you to the right people. For example, if you do lung and there are other people who do lung cancer, we're happy to serve as connectors and ambassadors for the Tumor Cell Atlas. Thank you very much for your attention. I would like to give a huge thank you to the START team. We're very excited to start our START project. And to all the patients and their families who contribute their samples. And to you for your attention, thank you. Even though I was not wrong.
Fantastic, thanks Arit, and thank you for stepping in slightly early. So we've got time for some questions.
So I was very intrigued to hear that you're shipping samples around to compare data sets. This is something we would like to do also in Human Cell Atlas Asia, among a few Asian countries. But then you always think that you're kind of founder on the rock of frozen tissues being freeze-thaw and damaged and so on. So are you doing that on intact cells, or are you doing it on nuclei when you ship the frozen samples?
So in terms of shipping frozen samples, we have many collaborators across the world. I think Partha is here, so even people from India came to practice these methods at the Broad. So for shipping samples, we have a very strict SOP for how to freeze the sample that you actually want to ship. This really helps if you have prospective samples and you can follow protocol and it's very clear directions. And when we've been doing that, we've had quite a lot of success. For example, Kristen, the GTEC samples, some of them are 5 to 6 year old being frozen, and they do very, very well. So I think overall, freezing frozen material for nuclei is good. For looking at cells, I think you can mostly only do immune cells and not, we haven't been successful of running single cell.
Makes sense, that's what I thought.
Yeah.
Thanks.
Thanks, Orit. Very, very, very interesting. Great start. I was wondering, this is in contrast to the normal, sort of to the organ-specific atlas. This is something like a layer on top of that, right? So, how are you planning to interact with the specific organ atlases? Is it even necessary, or for now you just leave it encapsulated in your samples, or would that be as a control, maybe a really interesting background?
No, I think we should interact all the time, right? Because you want to see what are the differences in the cell types, the cell states, the cell proportions. And it's always good to also have the expertise, the people who have the expertise in the specific organ, not only in the cancer, but in the normal organ. So I see this as a very integrated effort, and we do have cancer across most of the organs in our body, so I see it as something that would be very
So what's the first plan, the ICA, for example?
So, for example, if you look at the immune cell atlas, right, they will tell you that actually there's no, if they just look at normal immune cells, it's really not interesting and they won't find any important information. But if you challenge the immune cells, for example, in disease such as cancer, then you'll start really understanding what the function of immune cells is.
So the resistance is very, the immune-resistant tumors is very interesting. One question, which is, is this a pre-existing signature prior to the checkpoint therapy? And then the second, how does this relate to markers like PD-L1? So is it a distinct resistance mechanism and they don't need PD-L1 because they have an alternate mechanism?
So I think Aviv will answer better than me on this, but I think we can already see in some of the cells that the pre-existing resistant state. So we think it's mostly intrinsic.
Very neat.
Because the cohort that Orit actually showed was sampled I'll answer two things. Orit showed one cohort, but there are actually two, and the cohort actually comes from Germany, from the University of Essen, and that cohort is biopsied before and then treated. So the outcomes is after, but the states of the tumor are from before. And so it appears to be intrinsic. Even at the single cell level, when you look at patients that are untreated, you see subsets in the population that have this behavior. They just increase in frequency much higher in patients that have been treated and failed. So it seems like from all types of approaches suggest it's intrinsic, and it's not related to targeted therapy. So we also have patients from a second cohort, which were some treated with immunotherapy and some treated with targeted therapy, and this seems to only have be relevant to immunotherapy, but not to targeted therapy. Yeah, it's quite interesting in that sense, because there can also be acquired resistance, but it's probably through a separate mechanism than this one.
Hi, I'm just wondering from an organizational perspective, how are you guys dealing with different stages of diseases, like primary tumor, pre-cancer, metastatic disease, etc.?
So that's a very good question. We have different efforts. I think it's better if it's more systematic. Currently, it's not completely systematic. For example, for breast cancer, we mostly look at metastatic breast cancer because we have access to a huge amount of metastatic breast cancer tissues, while in colon cancer, for example, we look at untreated, mostly untreated samples. So I think that's a great suggestion overall, and it needs to be a more systematic effort to look at the difference.
Thank you.
One more question.
So I was wondering, when we look at the tumor, and like let's say someone suggests about the lung, that looking at the different part of the lung to make a complete map of the lung. So do you think, will it be possible to do something like that in tumor, to make a, look at the different part of the tumor? Because like in some patient, tumor will be occurring in one part of the organ compared to another.
So yes, so when we look at, for example, for GTEC samples, when we look at samples from organ donors, from deceased donors, either they're organ donors or rapid biopsy, we can really get hold of the whole lung, and then we can do a much better mapping. While when you look at live donors, it's usually a specific area where it's either biopsied or resected, and it's usually not the whole organ. But maybe Dana wants to say something about that? No? Yeah. So yes.
The data about the nuclei buffer having an effect, I was wondering if you were able to know if that's about the liberation of the nuclei, like is it differentially allowing nuclei out of the tissue, or does it change the way that they are lysed? Let's say, I'm assuming that was 10x or drop-seq. So is it affecting their ability to be processed downstream, or just which nuclei come out of the tissue during the, I guess, downstream homogenization or whatever you're doing to liberate them?
I think it's which nuclei come out of the tissue, because different tissues are very different. Some are more fibrous, some are less, some are more of a mesh. So I think it's that, because you can use different buffers and run on 10x on or whatever droplet or plate technology that you have, and you would actually get with buffer A good results with tissue C, and so I don't think it's necessarily the method that's what you're saying.
Okay, thank you.
Thank you very much, Arit.